division of the cell, the splitting of the cell, the equal division of the chromosomes. This is great for all the somatic cells, but as I mentioned early in chapter 10, doesn't really have anything to do with gametes. So here, with chapter 11, we're going to be looking at what it takes for the gametes. How do we go from one cell to multiple cells so that we can have haploid gametes? We need gametes for what we refer to as sexual reproduction. Sexual reproduction is when one haploid cell meets another haploid cell. They fuse. The two haploid cells make a diploid cell, a new diploid organism. This resulting cell, this resulting daughter cell, is genetically unique. A mix of the genes that were in the sperm, mix of the genes that were in the egg, come together, form something new. Look at you. Take you, for instance. You kind of look like your mom, kind of look like your dad, but you're unique. Now, keep in mind, this sexual reproduction, not all organisms out there have to do this. Some of them are going to go through what is referred to as asexual reproduction. Keep in mind, sexual, asexual here does not refer to what we think of as coitus, okay? It is not the physical act. It is the fusion and the mixing of the genetic material of two gametes. Asexual reproduction, this is going to be for pretty much all single cell organisms. This is how they reproduce. Binary fission or some version of mitosis. One cell splits, becomes two. So for sexual reproduction, two cells have to fuse. Now these cells have to be special. Okay, can't be any two cells in your body fusing together. Because any two cells in your body, chances are they're going to be two diploid cells. Remember, di means two, ploidy, chromosome set. So it's two chromosome sets fusing together would end up giving you a tetraploid cell. Tetraploid cell cannot function. Okay? It just will not work. Because then, two tetraploids fuse, you get 16 ploidy or 8 ploidy and 16, you get all sorts of weird numbers. So, hundreds and hundreds of millions of years ago, we slowly developed the process of forming gametes. Gametes are cells that are haploid. They have only one set of chromosomes. For us humans, that's 23 chromosomes. Now, throughout this chapter, we're going to find that why you look a little bit like mommy, a little bit like daddy, a little bit like your grandparents, but not complete like a clone of any of them, is that complete set, chromosomes 1 through 23, that you got from mommy. Well, which chromosome 1? Which chromosome 2? The one that she got from grandpa, the one she got from grandma. Same with 2, same with 3, all the way through to the 23rd. So you start mixing, matching. And then you start getting, same thing with your, the sperm you got from dad. Which chromosome came from, the chromosome came from grandma, came from, and on and on. So what we find is here, when we're talking meiosis, instead of giving you two genetically identical daughter cells like we saw in chapter 10 with mitosis, here meiosis is going to give us 4 genetically unique cells. Now, these daughter cells each will have 23 chromosomes, so that way they're haploid, so when they fuse with another gamete, you'll get a diploid offspring. But because it's going to be a mix of chromosomes 1 through 5 maternal, chromosome 6 paternal, chromosome 7 through 12 maternal, and back and forth like that between the 4 gametes that will result from meiosis, that's why we say they're genetically unique. Now, the machinery we talked about in chapter 10 for prophase, prometa, meta, ana, telophase, and the cytokinesis. The machinery we talked about there is also going to be in play here. So you're going to see a lot of the same enzymes, a lot of the same protein complexes. Many of the pictures are going to look the same. The main difference here with meiosis is that there's going to be 2 stages of division. Meiosis 1, well, it's going to look quite similar to mitosis. You're going to end up with 2 cells. Now, here right off the bat, the 2 cells are not going to be genetic copies of each other. Okay? The uniqueness starts here. In meiosis 2, you're going to end up with 4 gametes. All starting from one single cell, you end up with 4. It's a big difference. And when you line it up side by side, what you see is that, the artist draws it out. It looks a lot like the same stuff. Differences will start here in meiosis 1. Okay, in mitosis you had the two sisters. They would line up together, form the metaphase plate, as you see right here, the metaphase plate. The difference over here is this right there. The 2 sisters and the 2 sisters still get made during S phase, but instead of lining up like this, as they did in mitosis, they're going to be joined like this, 4 across. They refer to that as the tetrad. Meiosis 1 is going to separate the homologous. Meiosis 2 will then separate the sisters. So that as you progress down through, while the steps may look similar to what we talked about in mitosis, what's going to be different is this resulting haploid daughter cells. 4 of them, to be exact.